Hi, my name is Owen Leddy, and I'm a recent PhD graduate from MIT Biological Engineering, co-advised by Professors Brian Bryson and Forrest White. I'm going to give a brief overview of our JEM paper entitled Targeting Infection-Specific Peptides in Immunopeptidomics Studies for Vaccine Target Discovery. Mass spectrometry-based immunopeptidomics provides a proteome-wide strategy for discovering antigens that are presented on MHCs by cells infected with a pathogen of interest and could be recognized by T cells to confer protective immunity. We've previously applied this strategy to identify potential antigens for vaccines against tuberculosis, a bacterial infection caused by Mycobacterium tuberculosis, or MTB, that causes more deaths per year than any other human pathogen. One limitation of this method, however, is that untargeted mass spec methods end up identifying mainly self-antigens. In our prior study, MTB antigens made up less than 0.1% of all identified peptides presented on MHC class 1. In this study, we developed a workflow called PathMHC that can help more selectively target pathogen-derived peptides in immunopeptidomics studies. In this workflow, we split the MHC peptide sample into two parts, and first carry out an untargeted mass spec run comparing infected cells to an uninfected control. We then computationally identify peptides that are present in the infected cells and not in the control, and were not selected for isolation and identification during the mass spec run. Finally, we carry out a targeted follow-up run on the second half of the sample to identify these peptides. This lets us achieve greater sensitivity and prioritize peptides that are specific to infected cells. We first benchmarked this method by analyzing MHC class 2 peptides from dendritic cells that had phagocytosed a non-pathogenic mycobacterium, M. smegmatis, using this as a model system. PathMHC identified nearly twice as many unique bacterial peptides as did a comparable method based on untargeted mass spec alone, since our targeted analyses were enriched for bacterial peptides. We then applied PathMHC to identify MTB peptides presented on MHC class 1 by dendritic cells, and found that our targeted runs identified MTB antigens that our initial untargeted analyses had missed, here highlighted in blue. This expands the space of possible targets for TB vaccines. The MHC class 1 peptides we identified in this study exclusively derived from proteins exported by a specialized class of protein secretion systems called type 7 secretion systems, highlighting this class of proteins as promising targets for CD8 T-cell priming vaccines. Finally, we showed that our computational pipeline can generalize across multiple other pathogens by analyzing publicly available immunopeptidomics datasets from cells infected with SARS-CoV-2 and Listeria monocytogenes. We hope that this method will be a useful tool for future vaccine antigen discovery efforts.